At the Institute of Molecular Systems Biology, we investigate the complexity of biological systems. Specifically, we focus on the human cell. Each human is composed of around about 30 trillion individual cells. Each of these cells, in turn, contains at least millions of different biomolecules. These biomolecules interact with one another to carry out the function, whereas the most important class of molecules are the proteins. Dynamic protein organization, in order to regulate their function, is something seen in every form of life, like, for example, this piece of wood. And I would like to use it in order to give you an example. These are the proteins inside a human cell. And this one guy here is CSN5, a protein known to be involved in transcriptional co-activation when alone. However, he does not only occur alone, he also occurs in the context with his binding partners assembling into the COP9 signalosome complex. However, when in complex, he has an entirely different function regulating protein degradation. Now, we want to understand these principles and how the function of proteins are regulated on a global scale. However, we cannot see it, it's too small. Therefore, in this thesis, we developed a technology to not only measure the components of a proteome, but also their arrangement into protein-protein complexes. The technique is called Complex-Centric Proteome profiling. Imagine we want to measure the arrangement of these proteins inside the cell. And the trick now is to line them up by size. And lined up by decreasing size, we can now quantify each of the components by SWATH-MS spectrometry and generate quantitative signals. From these signals, we can conclude the composition of the underlying complexes. We extend concepts from peptide-centric analysis of SWATH-MS data to the level of protein complexes in complex-centric analysis. In this method, we quantify thousands of protein building blocks across the size range. To help interpret the vast amounts of data, we leverage prior knowledge on possible protein arrangements stored in public databases. With this method, we can characterize proteome arrangement on a systems-wide scale, detecting hundreds of complexes in a targeted fashion. We developed and distribute the computational framework for error-controlled complex-centric data analysis in an R package called CC Profiler. Now, the method can be employed to screen for functional rearrangements of the proteome coinciding with perturbations of the cellular system, like of the CCL2 cell line here, arrested in interphase and mitosis, an analysis that is ongoing. Remarkably, complex-centric profiling resolves and detects complexes in multiple different variants, like the COP9 signalosome complex and a putative new submodule visible here. Furthermore, the method can now be used to screen for functional rearrangements coinciding with perturbations of the molecular system. In the future, complex-centric profiling of the proteome will help elucidate new mechanisms of life. We are convinced that studying the proteome, including its context and the arrangement of its parts, will make great contributions to understanding how complex phenotypes, like for example complex diseases, actually emerge.